Hey class, welcome to my mitosis and meiosis test review. I thought I would make this video to sort of finalize y'all's understanding of mitosis and meiosis. I want to do that a couple of ways. Number one, I want to start out by talking to you guys about what the difference between the two are. Then I'm going to go through mitosis, and then I'm going to go through meiosis, and then I'm going to wrap up with some final thoughts. So I'm going to try and make this a fairly short video. I want you guys to follow along on your sheet so that you guys can stay engaged with this video, but just sort of fill in the blanks and sort of write whatever it is that I write. So first of all, I've got a couple of cells up here in the top left and the top right of your screen. That for one on the top left, this is in metaphase, which we're going to talk about in a second. And then we have this over here is in anaphase, because those the way I remember that is that those chromosomes right here are getting broken apart. So apart is anaphase, and then in metaphase, the chromosomes are lined up in the middle. So middle is metaphase. So let's talk about what the heck is the difference between mitosis and meiosis. Mitosis's goal is to create exact copies. That's the only goal that it has. We want to make exact copies of those cells. The reason we want to do that is because mitosis is going to create somatic cells or body cells. Body cells, you want them to be the exact same as the one before that. So your skin, you want your skin to continue looking the same. Your muscles, you want to continue your muscles to be the same. So body cells, we want to create them as exact copies. Meiosis, on the other hand, is the two goals of meiosis is to create genetic variation. This is important because genetic variation is going to allow more chances for your species to survive. One example I use is if chickens reproduced asexually, then if a yellow chicken disease came along, it would kill all the chickens. But because there is some variation in chickens, when a disease comes along, it doesn't kill off all of the chickens. The other main goal of meiosis is to have the chromosome number. Having the chromosome number is important because meiosis is going to create germ cells, which is like sperm and eggs. And so you want the sperm to have half the chromosome number and the egg to have half the chromosome number, so that when you put the two together, you're going to create a full, working organism. So let's first talk about mitosis. So the goal of mitosis, as I said, is to create exact copies. This is called asexual reproduction. But asexual reproduction is reproduction in which exact copies are created. Mitosis is going to do this in 4 phases. Hopefully you know this already. Some of you guys learned this in middle school, but basically the 4 phases are prophase, metaphase, anaphase, and telophase. Okay, those are the 4 phases. And one way to remember this is I use the acrostic, or is that an acronym? I don't know. One of the two. PMAT. So PMAT stands for prophase, metaphase, anaphase, telophase. We're going to dive into each of those phases here in just a second. So the 4 phases, prophase. I want you guys to draw these. So with prophase, what we have started to create are these things called centrioles, okay? They haven't migrated to the poles of the cells yet, but these are called centrioles, okay? There's some vocabulary here that I want you guys to know. We got centrioles. The nuclear envelope starts to disappear, and our chromosomes start to condense. So if you remember, these chromosomes are already copied. So this side of the chromosome is the same as this side of the chromosome. So this is 200% DNA. This side of the chromosome is the same as that side of the chromosome. So our chromosomes start to condense. Maybe you ought to label those. Sorry, I didn't mean to do that. These are chromosomes. And if you have a cell that looks like that, then it's going to be in prophase. Secondly, in metaphase, our nucleus has completely disappeared, and our chromosomes have lined up right in the middle of that cell. So our centrioles are over here. and they start to shoot out their mitotic spindle, is what that's called. So we know what our centrioles look like at this point. Let's go ahead and label this as mitotic spindle. Guys, I apologize for the little bit of lag. Whenever I'm screen recording, it does lag when I'm typing and I'm writing this stuff out. So bear with me, please. We got mitotic spindle. We know what centrioles look like, and we know what chromosomes look like. So let's move on. Metaphase, middle. We already kind of talked about that at the beginning of our video. In anaphase, our chromosomes start to get pulled apart, okay? So let's draw my chromosomes like this, because our chromosomes were in the middle, as we can see at metaphase, and then these centrioles are going to pull those chromosomes apart. So you can see that they have separated here and here. They have come apart from each other, and that's because the centriole is literally pulling those ropes. That spindle fiber are kind of like ropes. They're pulling those ropes towards themselves. So these are our chromosomes. That's anaphase. Anaphase apart. Let's talk about telophase. Telophase is interesting because we start to develop something that looks like a little bit like a booty cheek. Okay, they call this and this is what I want you guys to label, cleavage furrow. Now, if you can picture like one tennis ball or one water balloon would be a better example, getting pinched in half. That's what's happening. This is like a full sphere getting pinched in half by these ropes. Our nucleus starts to develop again, and our chromosomes are inside the nucleus. So telophase, if you see a cell getting pinched in half, that would be telophase with that cleavage furrow. And then finally, I know that the cytokinesis is not included in PMAT, but the fifth and final phase of mitosis is to finally separate those two cells. So we now have two cells here, and these are exact copies. Okay, so we want to remember that in mitosis we're creating exact copies. They look exactly the same. They're going to function the same. They have all the same DNA. We started with one cell. We now have two cells. We have successfully reproduced those cells. A couple last thoughts on mitosis. Mitosis is considered asexual reproduction. If you remember at the very beginning of the year, we talked about asexual and sexual reproduction. Asexual reproduction produces exact copies. Mitosis reproduces somatic cells. So meiosis produces body cells, things like skin, hair, nails, things like that. All right, so good job. We're done. If you can know all of those things, then you're good to go. Let's talk about meiosis, okay? Meiosis has two goals. Number one, we want to half the chromosome number. Okay? By having the chromosome number, we make way for germ cells, such as sperm and eggs, to be able to provide half of the ingredients necessary to make a new human or a new organism in general. And the other goal is to create genetic variation. This is what's different about mitosis and meiosis. In mitosis, we do not want any kind of genetic variation. We want those genes to be the exact same. In meiosis, however, we want to have genetic variation. We want differences, okay, to allow for higher chances of survival in the wild. So why is genetic variation so freaking important? There's my question. There's my answer. More chance for survival. That's basically it. I use this example every year. Let's just say one of my students has got webbed fingers. Okay, he was born with webbed fingers. He'd be kind of weird here at school. We'd be like, wow, that guy's got webbed fingers. But imagine if the whole world flooded one day. The dude with the webbed fingers is going to have more chances for survival, and the human race would continue to exist. If he didn't have webbed fingers, then the human race would all die off because we'd all die in the flood. So having genetic variation improves our chances for survival. How does it introduce genetic variation? There are two ways that we have genetic variation in a meiosis. Does two things. Number one, it does something called crossing over. Okay, and the second way is with a thing called independent assortment. Crossing over and independent assortment. So hopefully you guys took notes over this. Crossing over is the first thing that I want to talk about. Okay, so crossing over. You guys should have one of these drawn already, but we have a cell with black chromosomes on the left and blue chromosomes on the right. Now in crossing over, these chromosomes are so close together that some of the blue chromosome is going to get attached to the black chromosome, and some of the black chromosome is going to get attached to the blue chromosome. All right. Let's just say the same thing happens for this side. We got a smaller chromosome here, but it switches that bottom leg, and it really would be better if it were closer together. But basically you can see that this part of the chromosome switched with this part. They traded places. Another way to say that is it broke off and swapped places. Okay, and what this means is that when you finally go through the whole process of meiosis, which I'm not as bent on you guys knowing about, in one sperm cell you're going to have two blacks. In another cell you're going to have two black chromosomes. In the other cells you're going to have two blue chromosomes in both of those. Now in crossing over, so you have a 50-50 chance. One sperm cell, half the sperm cells are going to be blue, half the sperm cells are going to be black. However, over here, because of crossing over, when you go through meiosis, you're going to end up with one of these legs of chromosomes, all right? And on this first example, you're going to have one black chromosome, two black chromosomes. The right-hand leg of these, okay, you're going to have a blue and black chromosome and a black and blue chromosome. So that's a little bit different. And then in the third leg, we'll have a black and blue chromosome, and then a blue and black chromosome. So those broke off. These are coming from here, that leg. And then finally over here we got a blue chromosome and a blue chromosome. So as you can see, there's a lot more genetic variation in this cell that had crossing over than in the cell that had that did not do crossing over. Independent assortment. So that's crossing over. Basically, I just want you to know that parts of the chromosome are breaking off and swapping places. If you can know that and identify a picture of that happening, then you're good with crossing over. All right, let's talk about independent assortment. Okay. Independent assortment is much easier of the two. Basically, all that's happening is we've got this blue chromosome here. We've got this black chromosome here on the left, and then we've got blue chromosomes over here on the right. All that's happening in independent assortment is those chromosomes, because they're so close, they can actually swap places. So now I've got on the top, I have the blue chromosome on the left and the black chromosome on the right. And then I've got a black chromosome on the left. Those did not swap places. So they did not swap places. And all this is happening just randomly, okay? So as you can see, when you compare these two sets of chromosomes, they just swapped places. Now, when you consider the fact that we have 46 chromosomes, lining all those chromosomes up in the middle and then swapping places creates a lot of genetic variation. So in the same way, when you go through the whole process, you have the chance at getting this. And without independent assortment, you've got two black chromosomes in one, two black chromosomes in another, and then you've got in the other one two blue chromosomes. Remember, we're halving the chromosome number. One thing to consider, we've got 4 chromosomes here, but only 2 in all of these. So by the time meiosis is done, we have successfully cut the chromosome number in half. Over here, guys, we have a little bit more we have a little more variation. So when it's finally done, the left leg is going to have a blue and a black chromosome. The middle left leg is going to have a blue and a black chromosome. The middle right is going to have a black and a blue chromosome. And then the other one is going to have a blue and a black chromosome. So as you can see, there's a lot more genetic variation happening over here with independent assortment. All I want you to know with independent assortment is that we have taken 2 chromosomes and we have swapped places. They just go to the other side of the metaphase plate. The blue one went to the right and the black one went to, the blue one went to the left side and the black one went to the right side. So they just swapped places. This is going to create lots of genetic variation. So last thoughts, the goal of mitosis is to create copies. The goal of meiosis is to create genetic variation and to have the chromosome number. Shoot. There are 2 ways to create genetic variation, all right? 2 ways to create genetic variation. One of it is crossing over. And another way is by independent assortment. As long as you understand that crossing over and independent assortment both create genetic variation, then you're good to go. All right, and this last question before we end, this is your paragraph question that you're going to have on your test. Why would you want to repair the cuts on your skin with mitosis instead of meiosis? In short, the answer is because with mitosis, we want that skin to be repaired with the exact same cells that were there before. If we use meiosis, we would have some variation in the skin cells. So let's just say you have smooth skin in the place that you got cut. If you use meiosis, when that skin grew back, you may have rough skin or purple skin or blue skin, and we wouldn't want any of that. We want that skin cell to be the exact same. In this paragraph, I want you to sort of show me that you know what meiosis does, how it reproduces cells. Show me that you would want to say meiosis causes genetic variation using crossing over an independent assortment. However, mitosis produces exact copies of the cell. So I'm going to end this recording here. I know it's been long enough. I appreciate your attention. If you have any questions, feel free to let me know. Thanks.